Okay, this is how you can go to my website. You can use kenmitton.com or kenmitton.wordpress.com. We have also linked in staff tools, but also right on the main page, you can access our genome-wide map of where RNA polymerase-II is bound, basically in mouse photoreceptor cells in vivo. And looking between a couple of ages, you can see basically if there's RNA polymerase on a gene before photoreceptors mature versus after they mature. And that basically lets you figure out what genes are coming on in photoreceptors. And the before time is age P2, and the after mature stage is age 25 days. A lot of genome-wide data is always available in the geodatabase, but unless you use those software and tools to process either array scan files or DNA sequencing files and have the tools to do it, you don't really have access to the data. So what I've done is actually made my data available in the web browser. So if you do go to this part of my page, you can actually just click here, and it will launch and feed some of our data tracks. I've programmed them so you can just have them fed automatically into the genome browser. So this is the University of California Santa Cruz genome browser, and it's loaded in our track data. It will always open up and start automatically at the rhodopsin gene. That's a good gene to start on. One thing I recommend when you get to here is, depending on how big your screen is and how your font sizes are set, things might be easy to read or not. You can go right away, however, into the configuration button at the genome browser site and just make a few changes just to make life easier to look at. Your text size, you can put maybe up to 14. If you have a wider screen capability, you can probably put 1200 pixels wide. The labeling area for some of the tracks, you can maybe set that to 24. And just resubmitting this again, it's going to redraw the same view we had. But things are just a little bit easier to read now. Now there's a lot of tracks that you can turn on or off down with all of these various controls in the genome browser, and you can play around with them if you want, or you can just leave them alone. But basically the data we have here on the rhodopsin gene, if we look down here, here's the USC genes database listed, and you can see this is the structure of the rhodopsin gene. These little chevrons say that we're looking at the gene going this way. We're in this part of the chromosome. You can see it's chromosome 6. This way, from left to right, you're looking at the top strand. Genes that encode on the other strand would have arrow chevrons pointing the other way. The thick regions here are exon sequence. That sequence will all become part of the mature RNA. And within exons, there's a thicker and then a really thick band. The thicker bands show you where amino acid sequence is encoded, and the thinner areas are non-coding sequence of that RNA. So here's the 5' non-coding region, and here's some 3' non-coding region. What you also will see in here is Pol2 binding tracks in orange for age 25 and age 2. Now rhodopsin is not active at age 2. It's certainly active at age 25. It's the highest abundant messenger RNA in the retina, and it's the most abundant protein in the retina. And this orange band just tells you that our data picked up a significant amount of RNA polymerase binding in these regions. And it only happens at age 25 when the gene is on, because that's when RNA polymerase will be docking just in front of exon 1 at the promoter, proximal promoter, and transcribing RNA. And so it gets stuck to the DNA at different points throughout the gene. The blue bands up here, the blue tracks, will each show you a single line, which basically just tells you where we have RNA polymerase mapped in these genes. A peak signal, the most polymerase occurred here, and another peak occurred over here. We don't show you the data in little mountain range collections, but basically that's kind of what they look like. Now if I click on this when I'm viewing it, it should redraw it and actually have a little number beside it. This number is basically the signal strength that we got. And in this case, it's nice to compare to something like rhodopsin is a highly expressed gene in most photoreceptors, rod photoreceptors. So 16 is a pretty big number. And here there's a good peak as well, caught leaving the protein. None of these peaks show up in the P2. So you can see the P2 track is really clear of signal. And that's because in immature photoreceptors, while most of the rods exist in the age P2 most retina, they're not mature rods, and NRL has not docked on and activated the rod-specific photoreceptor genes such as rhodopsin. We publish this in Molecular Vision. When you click on any one of these tracks, you can also get more information on just annotated information and data that we've included. And I think if we click, let's see, over here now, let's click on a band. And I think if I click directly on one of the bands, oh, maybe not. I want to get to the little annotation data that we have. Oh, it's just added them here. Added their label. When I click on the label, that brings me to some information that will be specific for that little labeling piece of data. It'll also give a little bit of description of what this experiment was about. And you can see down here a couple of papers that we have done based on data. This one in 2010, Molecular Vision, tells you all about how the data was generated to map RNA polymerase. Now the handy thing about this database being in this format is you don't have to be a programmer. You don't have to get the original bar files that we got from mouse promoter tiling arrays. You don't need to get software or know how to process them to look at the data. You have a nice visual representation here. So you can look for any gene of interest. So, you know, if we looked at something like PDE 6B, now say this was a gene you didn't know anything about, or you were wondering if it comes on in mature rod photoreceptors, you could basically select it. You could go to that gene, and you can look to see if there's a little or no RNA polymerase at age P2, and if there's a lot at age P25. Now, of course, for phosphodiesterase 6B, it'll be like rhodopsin. There's really no RNA polymerase in the promoter region of the gene. So again, I'm zooming out 1.5 times because I want to see in front of exon 1 or right around exon 1. You can see in this gene, like rhodopsin, at P25, there are tracks, lots of tracks being picked up for the presence of RNA polymerase. Nothing happens in the P2 data set. And again, just for the P25, there's large peaks where large amounts of RNA polymerase were mapped. And in P2, immature retina, nothing. So if you have a gene and you want to know if it probably is enriched in photoreceptors as they mature, especially rod photoreceptors, if you see this kind of pattern, this would predict that it is. And as a matter of fact, in this data set in the paper in Molecular Vision, we found 800 genes activating in rod photoreceptors that had not previously been described by DNA sequencing, probably because it wasn't done deep enough at the time, or by expression microarrays. So this is a very handy tool. Some other groups have used it. If you find there's data in here that you want to include in one of your papers and you would like to have the highly detailed mapping mountain range versions of where all the RNA signal is, I'm happy to provide them to you. all you have to do is email me, Mitton, M-I-T-T-O-N, at oakland.edu. I've provided that data to a couple of other labs that have used them in their papers, because this is supposed to be a public science resource, and that's how you can get an idea of what comes on genetic-wise in a rod photoreceptor as it goes from immature to mature active state, all ready to detect light in your retina. Thank you.